for the opportunity to present some of my research. So first of all, I start with like a mission statement of the work that we do, that is working at the interface of physics with chemistry, material science, and biology. And we work both in nanomaterials and biomolecules. I will not focus on the nanomaterials work today, but only on the biology. But again, we have other work there that I would be happy to share some other time. And we mostly work with problems that are inspired by experiments, using a range of methodologies, so from ab initio to classical molecular dynamics, and also an unsampling method, depending a little bit on what is needed for the particular problems. And if we want the sort of common thread of the very different problems we sometimes studies, is that we look a lot at how particular system react to external stimuli like light, pressure, the binding of a ligand, and similar. So concerning the biomolecules work that we do, we've been working on polyphenols interacting with various proteins or DNA. And we have been working on neuroreceptor, that is the topic I will mostly focus. Neuroreceptor also can be activated by specific molecular switches that are also common to other systems, for example, protein aggregation and fibrillation development. And this is another problem we have looked at. And then we've been working on fluorescent probes and photo switches that again links a little bit with the molecular switches. So some of the problems seems quite different, but they have common techniques and common actually ideas, and this is actually interesting. So I said that I will focus on the neuroreceptors and their environment and what are the challenges, and also the opportunities that are available in these at these times with the development of new experimental methods and also new computers and new techniques and so on. So we have a lot of nerve cells, the neurons in the brain, and these are connected through again many connections that are called synapses. And the neuroreceptor we're interested in are linked to the synaptic communication, and so are linked to these sort of red spots between the various neurons. So the way the signal, the electrical signal is somehow passed is that some neurotransmitters, some small molecules are released, and then they travel through the synaptic cleft, and they go to activate these neuroreceptors that are embedded in the cell membrane. They have a pore that is closed until the neurotransmitter attached to the external part, the extracellular domain of the neuroreceptor, and these activate a series of structural changes that culminate with the opening of the channel. And so the ion then can pass through and so change the cell potential and transmit an electrical signal. Now, obviously, these receptors are involved in the communication between the neurons. So they are involved in many neurological disorders, from Alzheimer's, schizophrenia. They are involved in, they are the target of anesthetics, for example, drugs like benzodiazepine, but also in insecticides and so on. So they're very important, but they're very complex nanomachines, and we have little idea of how these ligand-gated ion channels, because okay, there are ion channels and they are activated through the binding of ligands, act and behave at the molecular levels. So the question obviously is whether we can help by using atomistic simulation to get some ideas at least of how the ligand-gated ion channels function at the molecular levels. And we wish that this is true. So we can actually make models, atomistic models of these receptors now. And this is on the right is a representation of one of the prototypic or representative of these pentameric ligand-gated ion channels. So there are 5 subunits that are arranged around an ion permeable pore. A part is outside the cell membrane, and the ligand would bind in between 2 subunits. And then we have the transmembrane domain that is made up by alpha helices, 4 for each subunits. They're labeled M1, M2, M3, and 4, with M2, the one that line the channel, and M4, the one outside. And this part, the interface between the 2 domains is very important because is the area where the signal is transmitted. So what are the challenges? So the challenges are where and how does the neurotransmitter bind, and also how does it unbind? How this binding translates into an opening of a channel that is quite some tens of angstrom far away. Until recently, I just had these 2 ones, but actually there are others. One is the role of the membrane. Is there an active role of the membrane, of the composition of the membrane, and so on? And then looking a bit more in the future is can the opening of the channel be actually manipulated in some way, not just with different ligands of drugs, but also with light, for example. There is all a big optogenetics area that is very interesting. And there are also very new interesting opportunities because there are new experimental structures by cryo-electron microscopy and X-rays. Until very few years ago, there were basically no structures. There are new data, and of course there will be new computer power and lots of computer power with exascale computer in the future. But also it's not just a matter of brute force, but also of improved and novel ideas for computational techniques. So let's see what we have done so far and what we are doing at the moment. So I will first concentrate on some work that we have done on binding. So in the next slides, where I just represented the 2 subunits for the extracellular domain, and through metadynamics simulation, we actually evaluated and mapped the free energy landscape of binding of GABA in this receptor. Again, is another one of this family that is related, is an insect receptor that is related to insecticide resistance. And we can map actually, but just to say something, that they are all very similar structure. And so basically the methodologies, sorry, let me go back 1 slide. So the methodologies and the protocols can be used actually also for other members of the family. So in this case, we actually map the free energy landscape, and we identify, for example, some mechanism where this particular loop in orange open and close like a cat flap. And also we, this is a projection on 1 dimension of the free energy landscape with the bound, the pre-bound and unbound region. And we saw how this is disrupted by particular mutation that were actually tested experimentally in Sarah Lammy's lab at Cambridge, and that produce non-functional channels. And we're interested now in actually binding and unbinding kinetics, in particular because it's possible to measure them, and the group of Lucia Sivillotti in the pharmacology department at UCL is doing single channel measurement with that, and we started actually looking at this challenging kind of simulation for determining kinetics of binding and unbinding. We look at other issues and other problems. This is another receptor that is related to the vomiting center and binds serotonin. And we were interested in a potential molecular switch at the interface between the extracellular and the transmembrane domain that has been identified experimentally and consists in a proline that can isomerize for trans to cis. If this does not happen, or cannot happen, and this was tested through mutation, then the ions would not flow. And so the system would not function. And this, at the beginning, we actually could just study the switch, so proline dipeptide in water, that is a very small system. We studied it with metadynamics and considering various mutation and so on. But we are now in a position to study actually what happens in the Wool receptor and how this energy landscape is actually changed by the environment, but also how then this switch, this potential switch, which is still quite controversial, actually affects the receptor itself. And here on the right, there is a representation of the interior of the channel and how actually isomerization of this particular potential switch modify actually in particular the selectivity filter for the ions. We're interested at the moment we got interested at the moment in the membrane. So again, the first simulation I showed, we didn't even consider the membrane. The second simulation I showed, we just consider a simple membrane with a single lipid, but actually the membrane of cells is much more complicated, and there's very recent interest in that, and there's still no conclusive data whether lipids actually can modulate the response. So what we have done so far is to consider 3 different compositions with different phospholipids and a different percentage of cholesterol, and see what actually happens. And another project that we are at the moment is that we're looking at the function of the external helix, the one at the interface with the cell membrane. And again, this is studied in Sarah Lami's lab in Cambridge. Now, putting more cholesterol in the membrane increase viscosity, and with this, I just want to pass to another project that we're looking at that is not directly related to neuroreceptors, but is actually related to how one can measure and image viscosity in a complicated biological environment. So the cell actually is very viscous. There are different regions of different viscosities, but the change on cellular viscosity can also be a sign of disease, and many diseases actually are related to viscosity. One can think about hypercholesterolemia, because more cholesterol obviously make the cell membrane more viscous, but also cataract and so on. And so how does one measure or image viscosity in this, in vivo, for example? So one possibility is to use molecular rotors, and this is a prototypical one based on body P, where again, there is a die with a part that is this sort of paddle that can rotate, and this rotation can actually change if the the environment is more or less viscous. And it's possible to map the fluorescence lifetime with the viscosities. These is work done in Klaus Sully's lab at King's, and on the other side, and then it's possible actually to use fluorescence lifetime imaging to image actually the viscosity of cell. And these are some other work done in Marina Kulmova lab at Imperial, where again, the cell membrane of eye lenses, the eye lens is actually mapped. So the issue is how does this molecule work? And so we can actually try to help to understand this by doing a series of simulations. So first of all, we can do some static calculation to map the free energy, the potential energy for ground state and excited. The first excited state is mostly homo-lumo kind of excitation as a function of the angle, and we can start looking at some potential mechanism of excitation and de-excitation. And of course, if the rotation is hindered by the solvent, there might be different kind of behavior. But static calculation might not be enough because there are various movement, not just the rotation of these, but also, for example, swinging motion and so on. So temperature becomes important, and actually some of these rotors with various functionalization are actually maybe not viscosity sensor, but can also be temperature sensor or polarity sensor and so on. And these are some simple calculation done with the simple excited states method that cater for homo-lumo excitation. And then, okay, we can put the environment in a QMMM method. We study methanol and glycerol, and also then go towards completely classical simulation, but with very complicated environment to help understand some of the classic of the interaction of this molecule with the environment that are studied experimentally. So just to conclude, we can actually use atomistic electron structure simulation to actually try to understand very complicated nanomachines like neuroreceptors, but also to try to understand better their environment by helping also experiments to interpret and give some atomistic and electronic structure information that are useful to experiment. We cannot just use a single technique, but also technique at different levels of resolution. And it's an exciting time because, again, there will be much more computer power that would allow, again, much more sampling, but not just brute force calculation, but also improvement in theory, try to look more about innovative methods like an unsampling method to look, for example, at things like kinetics and so on. And then again, there are lots of ideas, for example, to, again, in the fields of optogenetics, to try to manipulate with the light, also the opening of the channels and also the communication between neurons. And so again, possible improvement would be better excited states method than what we use, better ways to include the environment, and so on. So I just want to acknowledge my group, and these are the students that are working with me at the moment on ion channels and on rotors, but also I want to mention the work on ice crystals and nanomaterials under pressure, and in particular we're using now nanomaterials under negative pressure that again I will be happy to speak about some other time, and the experimental and theoretical collaboration. And just to do a moment of advertisement, we have a new doctoral training center, King's on Biological Physics on Scales, and there are some openings. So if people are interested, please look at the website or ask me. Thank you very much for your attention.